Now that we've had an overview of cellular respiration, let's take a look at each of the steps in a little bit more detail. At the end of each step, we want to be sure that we summarize what has happened in each step, and specifically the status of our carbon atoms, how many ATPs have been generated, and also how many full electron carriers we have. And so we'll do that at the end of each of our steps. This picture shows a little bit more detail than our picture that we looked at earlier. You can see that in the process of cellular respiration, we're starting with glucose. And I hope that we remember that we have 6 carbon, 12 hydrogen, and 6 oxygen. Remember that in cellular respiration, we are going to be oxidizing glucose to carbon dioxide, which means we'll be taking our glucose and breaking those bonds and taking the electrons and the hydrogens and placing them in the electron carriers. I hope we remember that throughout this process we will generate some ATP. So 2 ATPs are generated in the process of glycolysis. What's not shown in this picture is that there actually has to be a small investment of ATP to get glycolysis going. So it's a good idea for us to at least note somewhere that there has to be an investment of 2 ATP molecules. Technically, glycolysis is going to produce 4 ATP molecules, but if we subtract out the 2 that we have to invest, then we're left with only 2 ATP molecules. So this picture right here is showing us the final result of our ATP. We invest 2 ATPs, we make 4 ATPs, and then we're left with 2 ATPs. So it's important when we read how many ATPs are made, it's a net of 2. I kind of think of this like a bank account. If you want to open a bank account, you have to have some money to put into that bank account to be able to go through the process of depositing, withdrawing in the future. And so you have to have some money to invest to start. We also know that we have to have incoming NAD+. So I hope we remember that this is our empty electron carrier. And it's good now to remember, where is this coming from? Well, at the end of our process, I hope we remember that all of our NADHs, those that are full, are going to go to the electron transport system, and they're going to drop off their electrons there, which means that coming out of the electron transport system, we have emptied out our NADHs back to NAD+. So these NAD pluses that we see here, those are coming from our electron transport system after those carriers have dropped off their electrons. So we can see here in glycolysis, we'll pick up more electrons. Technically, each of these carriers can carry 2 electrons. And so if we have 2 NAD pluses, then we can have 4 of our electrons being loaded, and of course our hydrogens as well. Our final product for glycolysis is going to be pyruvic acid. And if we just look at the chemical structure of it, you can see that we still have 3 carbons, and those 3 carbons are across 2 pyruvic acids. So you can imagine that we just take our glucose and we break it in half. We can still account for all of our carbons in our pyruvic acid. Let's summarize glycolysis. Regarding our carbon, at the end of glycolysis, we can still account for our 6 carbon. They are now all in pyruvate. There are 2 molecules, and each of those pyruvates have 3 carbons in the pyruvate. So that's the destination at the end of glycolysis for our carbon. We know the summary for our ATP is that we invested 2, so we used 2. We made 4, and that means we have a net of 2 ATPs that are going to be made. And finally, our electron carriers, our NADHs. So far, we have filled up 2 of those. That's the status of our full electron carriers. Glycolysis gets a little bit of the energy out of the original glucose molecule, but not very much. At this point, we only have 2 ATPs. So we have to continue along the cycle to make sure we can extract as much energy as is possible out of the glucose molecule. If you remember from our overview of cellular respiration, our next step is the grooming step, or the transition step, or sometimes termed the preparation step. This transition or preparation step is going to take us from our cytoplasm into the mitochondria. So I'd like to take just a brief minute as we move into the mitochondria to make sure that we're clear on the different layers of our mitochondria and exactly where we are in the cell. Out here is our cytoplasm, and we've already established that the process of glycolysis is going to happen there. We also remember from a previous discussion that our mitochondria have two layers. There's the outer membrane of our mitochondrion and the inner membrane of our mitochondrion. And this figure is labeling our different compartments a little differently than we have talked about these before, so I want to make sure that we're clear. This area here that they term the outer mitochondrial compartment is what we before have called this intermembrane space. So this is the same thing, and you can see it really nicely here. This is the space that's in between our inner and outer membrane. And what they're terming the inner mitochondrial compartment is the matrix. So that's the word that we used before. I hope that both of these terms are pretty easy to memorize. We're just describing the spaces in relationship to where the membranes are. Our transition step or our grooming step is then going to carry us from the cytoplasm into this intermembrane space. So you can see the end result of our glycolysis, our pyruvate then, is going to move into our matrix of our mitochondrion. And this arrow really represents what's happening then in that step, in this transitional or preparation or grooming step. So let's recap a couple of things about that along with the outputs. We know that our pyruvate has 3 carbons each. And through the process of our transition step, we're actually going to make this molecule here called acetyl-CoA. And so acetyl-CoA is the end result of our transition step. If you want to, you can even label this transition step or this grooming step here, because this is really representing where that's happening. Since each pyruvate has 3 carbons each, and we have 2 of them, each of them is going to lose a carbon and some oxygen in carbon dioxide. And that's ultimately then going to leave the cell. It's actually going to leave the mitochondria. And this is what ultimately we exhale. So keep in mind that when you exhale carbon dioxide, this is one of the places that this carbon dioxide is coming from. That means that each acetyl-CoA now is only going to have 2 carbons each. We are also going to continue to remove hydrogens and their electrons and load them up into electron carriers. So we can see that 2 more NADHs are going to be formed. That means that we had to have an input of our NAD plus coming from the electron transport chain, just as we had back in our process of glycolysis. And those NAD pluses are going to pick up electrons, and so we have 2 NADHs that are going to be formed. Finally, we can note that in our grooming step or our transition step, we do not have any ATP that's formed. So when we're identifying where this grooming step is, we have our 2 pyruvate coming out of glycolysis, and our results are 2 acetyl-CoA, our 2 NADHs, and our 2 CO2s. Those are going to be our products. It's important that we realize for our ATP production that no ATP is going to be made in this process of our transition or our grooming step. So no additional ATP is going to be generated at this time. So to summarize our grooming step, we know that regarding our carbon, let's find out where all 6 of our original carbons are. Well, for each of our pyruvic acid, we had 3. And we know that 2 of those carbons are going to be in a molecule of acetyl-CoA, and we know that 1 of those is going to be released as carbon dioxide. However, because we have 2 of them, 2 of the pyruvic acid, and therefore 2 of the acetyl-CoA, we know that that's going to all be duplicated. So that's the status of our carbon at this point. We've also established that regarding our ATP, there is no ATP, 0 ATP made in this step. And we also know that regarding our full electron carriers, we have made 2 more. We now have 2 NADHs. If we're keeping track of our total, we actually had 2 from glycolysis, and now we can add to it 2 from our grooming step or our transition step. Let's move into our next part, our third step in cellular respiration, and that's going to be our citric acid cycle. The citric acid cycle used to be known as the Krebs cycle, and that might be a term that you've heard of before. But this has now been renamed, and it's essentially where our citric acid cycle is. So whenever you see Krebs cycle, it means the exact same thing as our citric acid cycle. Let's talk a little bit about our inputs and our outputs of our citric acid cycle. At this point, the only thing that's going into our citric acid cycle, and you can see it because of this arrow right here, is going to be our 2-acetyl-CoA. So that's what's going into our cycle. We put in one and we turn the cycle, and then we put in the other one and we turn the cycle. So the outputs of what's coming out of our citric acid cycle here are the totals for putting in both of our acetyl-CoAs. And I hope this matches up to a picture that we drew together in an earlier question set. So let's make sure we understand what's coming out of our citric acid cycle. We know that we essentially finish the process of oxidation of glucose, which means that all of the remaining carbon is going to be lost. That means all of the remaining carbon is going to now be in a carbon dioxide molecule. So here we can see that 4 carbon dioxides are going to be released. If we add those 4 to the 2 that we released in our grooming step, we can see we've accounted for all 6 of our carbons. 4 of the carbons are here, 2 more of the carbons are here. So all of the original carbons that were in our glucose are now released from our mitochondrion and from the cell as carbon dioxide. We also know that all of the remaining electrons in our hydrogens are going to be loaded up into those electron carriers like we talked about. And as we enter into our citric acid cycle, there are still a lot of electrons to load up. In fact, when we put in both of our acetyl-CoAs and turn the citric acid cycle twice, we're going to get out of it 6 NADHs and 2 FADH2s. And we've already talked about the fact that FADH2 is just an additional type of carrier. It does the exact same job as our NADH does. So those are a lot of filled carriers. Finally, we need to account for our ATP, and there is some ATP generated. When we turn the cycle 2 times, the final result are 2 ATP molecules. It actually makes 1 ATP molecule per turn, so if we have to turn it twice, we get out 2. Hopefully we can see that that is our citric acid cycle summary. Our carbons are now all in CO2. We've made 2 molecules of ATP, and we now have 6 NADHs plus 2 FADH2s as our full electron carriers. We're now ready to move on to our last and final step of our cellular respiration process, and that is our electron transport system, where we're going to generate most of the energy that we can from our glucose molecule. We're going to look at a couple of pictures of our electron transport system. Let's make sure we're clear about where we are. The electron transport system is embedded in our inner membrane. This membrane right here is our inner membrane of our mitochondrion. This membrane here is our outer membrane of our mitochondrion. And then we remember that we have our intermembrane space here, and our matrix is here. So it's important that before we zoom in and look at a closer look at our electron transport system, that we're really clear about where we are in our mitochondrion. You can see that if we start at the beginning of our electron transport system, which is going to be here, we're going to be dropping off electrons, and the electrons are going to be moving through this electron transport chain. And that's indicated here, showing that our electrons are being transferred through our electron transport chain. We need to be sure we understand where these electrons are coming from. This is what all of our carriers have been doing throughout the process of glycolysis, our grooming step, and our citric acid cycle, is that they've been picking up electrons, and these electrons are now delivered to our electron transport system. So per glucose, we actually have a total of 10 NADHs. I hope we can see 6 here, 2 here, 2 here. That's 10 NADHs, and then we have 2 of our FADH2s, and those are being dropped off here. So a lot of electrons are being dropped off. Just like we saw in the light reactions of photosynthesis, an electron transport system is going to harness the energy of falling electrons. So as these electrons are moving down the electron transport system, hydrogen ions are going to be pumped across our inner membrane, and we're going to have a high concentration of hydrogen ions located in this intermembrane space. So that's where we're going to see a high concentration of hydrogen ions. And then we're going to allow hydrogen ions to flow down their gradient through an enzyme to actually make ATP. I hope that you could label this enzyme that we're talking about. It's not labeled on your picture, but we now know that this enzyme right here that's making the ATP is called ATP synthase. Same enzyme that we had back in the light reactions of photosynthesis. So the hydrogen ions are going to move down their concentration gradient from an area of high concentration to an area in here in the matrix where there's a low concentration of hydrogen ions. And as they pass through our enzyme, ATP synthase, this is when we're going to be able to take ADP and a phosphate group, and we're going to be able to make that ATP. And here is where we generate a lot. We said that the electron transport system makes upward of 28 ATPs per molecule of glucose. This is many more times ATP than we ever made back in glycolysis or our grooming step or our citric acid cycle. And so here is where we really get the energy from our glucose molecule by transferring electrons. We also need to see the special role that oxygen plays and where it works. We've established in a previous question set that oxygen is going to be here at the bottom or the end of our electron transport system. And so we know that oxygen is going to be the final electron acceptor. The final electron acceptor, which means that it's going to grab a hold of that electron as it falls down the electron transport system. And when that happens, we're going to be able to generate water. So when oxygen grabs those electrons, so we've got essentially O2, plus some of the electrons, plus some of the hydrogens, ultimately that's going to give us water. And so you can see that water is generated as a part of this process. Let's take a look at another picture that zooms in on our inner membrane so that we can make sure we're clear about this process. It's always helpful to look at a number of different kinds of pictures of the same process so that we can make sure we get this down. So let's make sure we get our bearings and understand where we are, because the orientation of this picture is just a little bit different than the orientation of our other. We know that our membrane here is the inner membrane of our mitochondria. We can kind of see that here. But in this perspective, the outer membrane would be here. So if you want to, you can sketch where the outer membrane would be. In fact, we have a number of things to label on this picture. That means that this space in here is going to be the area of our intermembrane space. And that means that the area here is the inside, the innermost side of our mitochondrion, which is the matrix. So understand that we're talking about a different orientation. It's kind of the flipped orientation from the previous picture. Everything's the same, it's just that we're looking at the outer membrane at the top of our figure rather than at the bottom of our figure like we did in the previous picture. So let's first find our electrons and see where they are and where they're being dropped off. Right here, we'll find that our electrons are being dropped off and starting to move through our electron transport chain. So let's first find out where our electrons are being dropped off. Right here, we can see that NADH is going to be dropping off our electrons at the very first of the proteins that are embedded in our electron transport chain. And when NADH drops off its electrons, this is where we're going to recycle that back to NAD+. So I don't want us to think about this as an afterthought. This is really important process. This NADP+ then is now empty, and where's it going to go? Well, it's going to go back to glycolysis to pick up more electrons, or back to our grooming step to pick up more electrons, or back to our citric acid cycle to pick up more electrons. So the action of dropping off these electrons at the beginning of our electron transport chain actually makes it possible to go back and keep glycolysis, our grooming step, and our citric acid cycle going. We have to have an ability to be able to empty out those carriers. FADH2 is also going to drop off its electrons. It just does it a little bit later here in our system. So you can see NADH dropped off here at the very beginning. FADH is going to drop off just at the second protein. And when that empties, we'll have FAD. And again, the status of that FAD is that it's going to go not back to glycolysis or the grooming cycle. FAD is only used in our citric acid cycle. So it's going to go back to the citric acid cycle and pick up some more electrons. These electrons, as we remember, as they are being passed down through our electron transport chain, we're going to use that energy to move hydrogens across our membrane. So let's label and find our hydrogens. We can see that our pluses here all represent these positive hydrogen proteins, and we can actually include them here with our molecule. As our electrons fall, our hydrogens are going to make their way across the membrane, and we're going to have a really positive area here in our membrane space because we have a high concentration of our hydrogen ions. Where here in our matrix, we're going to see a more negative charge because this is where we have a low concentration of our hydrogen ions. And that's the same gradient that we've talked about before. In this picture, we can also identify our ATP synthase. Let's find the place where our hydrogen ions are moving across our membrane from an area of high concentration to an area of low concentration, and we're actually making our ATP. And you can see that that's happening right here. So this molecule right here at the very end is our ATP synthase. So here it is, ATP synthase. And as those hydrogen ions move through, then we're going to be able to generate a whole bunch of ATP. And we said upwards of about 28 ATP as we allow for hydrogen ions to move. The last thing that we need to identify here on our picture is where our oxygen is and where we produce our water. So we need to look at the end of our electron transport system to find that. So right here we can see our oxygen molecule, O2. And when we capture those electrons that are falling down the electron transport system, then we can make H2O, and that's what this molecule is here. Now, I haven't really balanced our equation. Technically, it's half of an oxygen molecule, which is just an oxygen that's going to grab a hold of two of our hydrogens to make one molecule of ATP, but it's okay if we just recognize that we have oxygen there at the end of our electron transport system to make our water. Remember that this oxygen that we're talking about right here, this is the oxygen that you and I get from breathing. So moment by moment, as we're breathing, we're providing oxygen to our cells. More specifically, we're providing oxygen to our mitochondrion. And the only role that oxygen plays anywhere in your entire body is going to be right here at the bottom of the electron transport chain as a final electron acceptor. So this is the entirety of the reason that you and I breathe oxygen, to be here at the bottom of our electron transport chain to be the final electron acceptor. One of the terms that we used back in our light reactions that we can also see at play here is that word chemiosmosis. So the word chemiosmosis. And it means the same thing here as it did back when we talked about it in photosynthesis. Chemiosmosis is when we link together or couple together two things. And what we're coupling together in the process of chemiosmosis is the first thing being our creating a hydrogen ion gradient. So right here, when we use the energy of falling electrons to create a hydrogen ion gradient, we're going to couple that with ATP production in ATP synthase. ATP production and ATP synthase would not be possible if we didn't have a hydrogen ion gradient. And so because we do have a gradient that's generated by allowing our electrons to fall down the electron transport chain, we're able to make ATP. That's what chemiosmosis is linking these two processes together. Let's summarize the different stages of cellular respiration with the number of ATPs that are produced and where this is happening. I hope we remember that in glycolysis we produce 2 ATPs and that this process is happening in the cytoplasm. In our pyruvate grooming step, transition step, preparation step, we actually don't make any ATP. We're just getting ready to go into our citric acid cycle. And we know that that step, while it starts out in the cytoplasm, is really happening in the mitochondria. In our citric acid cycle, or our Krebs cycle, we're going to produce 2 ATPs total from our 2 acetyl-CoAs that go in. And again, that's happening in the mitochondria. And finally, in our electron transport chain, we're going to produce 28 ATPs, and that happens in the mitochondria as well. So we can see overwhelmingly that the vast majority of the energy that we transform from a molecule of glucose is going to be occurring in our electron transport chain deep in the mitochondria. The next issue that we need to address when we're talking about cellular respiration and energy is what would happen to a cell if it doesn't have oxygen. So I'd like us to think a little bit about this as we look at the big picture, because this will help us understand some of the other processes that we're going to talk about. Let's imagine that in this cell we do not have oxygen. Let's say it's not available for some reason. Let's think about what that means for our overall process of the electron transfer system. If we don't have oxygen, then our electron transport system essentially is going to come to a screeching halt. The reason for that is because if oxygen is not there as the final electron acceptor, then kind of like a conveyor belt that gets shut off, all of these electrons are going to back up this direction, and we're no longer going to be able to allow for those electrons to move down this electron transport chain. What that means is that our NADHs that are coming from all of the different steps we've talked about, as well as our FADH2s, they're no longer going to be able to drop off their particular electrons. And so they are essentially going to be stalled out and not able to empty out, which means that you can't bring back empty electron acceptors to our citric acid cycle, to our grooming step. So essentially, if you don't have oxygen, it not only shuts down the electron transfer system, but it also shuts down our citric acid cycle because we don't have any way to collect those electrons because there are no empty electron carriers available. We also can see then our transition step as we back up knocked out as well. So essentially, if there's no oxygen, our electron transport system, our citric acid cycle, and our grooming step all come to a halt simply because we're not able to empty out those NADHs. However, if you remember back to a previous picture we talked about, our big overview that we labeled in an earlier question set, there was one place where we had drawn an alternate route, a fork in the road. And on this picture, that fork in the road is going to be right here. If there's no oxygen, for some cells, and for some times in some cells, we can go down an alternate route, and that alternate route occurs if there's no oxygen available. I wanted to make sure we understand some terms about what it means if we don't have oxygen, and then talk about some of the cells and processes that can occur. If we have oxygen available, so in general, everything that we've been talking about with oxygen present, we would say that that is an aerobic process. That might be a word you've heard before. Aerobic respiration means there is oxygen and all of our stages are occurring. Glycolysis, our grooming step, our citric acid cycle, and our electron transport chain are working fine. And in fact, we even use this word when we talk about going to the gym and going to aerobics class. When you engage in aerobic activity, you're not working out so hard that you can't deliver oxygen. When you're doing aerobics or jogging at a slow pace, you're actually able to deliver enough oxygen to all of your cells so that you can engage in cellular respiration and produce the ATP that you need. However, if there's no oxygen available, then for some cells we can switch over to what's called an anaerobic process. So anaerobic means without oxygen. The types of cells that can carry out anaerobic metabolism are pretty few in number. I wanted to give you an example of a couple of different types of cells that can carry out anaerobic metabolism. Some examples that we're going to talk about next occur in yeast cells. So there are some yeast cells that can carry out anaerobic metabolism. There are some bacteria cells that can carry out anaerobic metabolism. And then there's even some cells in humans and other mammals, and those cells are muscle cells, so human muscle cells. Your human muscle cells can carry out anaerobic metabolism for a very short period of time, maybe between 30 seconds and a minute or so. And after that, they would no longer be able to produce ATP without oxygen. Let me tell you how you would know that. Let's say you decided to do an all-out sprint. I mean, running as fast and as hard as you possibly can, 100%. And I want you to think about how long you could do that. Even if you're in really great shape, great physical conditioning, you really wouldn't be able to do that for longer than 30 to 60 seconds. When you are all out running at 100%, you are simply not able to breathe fast enough or to get your heart to beat fast enough to be able to deliver enough oxygen to your tissues. And so you only can do that for a very short period of time. When we look at some of these different cells, there are a few different processes in aerobic respiration, and I'd like to talk about two of them. The first process that I'd like to talk about is lactic acid fermentation. This word, fermentation, is a word that we use whenever we're talking about an anaerobic process, so without oxygen. And the types of cells that can carry this out that we'll focus on here are going to be bacterial cells. And in fact, if some bacteria are limited for the amount of oxygen that they have, they'll take over this other pathway called lactic acid fermentation. Your human muscle cells are also carrying this out when they're denied oxygen, like we talked about moments ago. So let's look at what happens in lactic acid fermentation. We start out with glucose, as we always do at the beginning of glycolysis. And in fact, glycolysis is going to proceed as normal. So whether you have oxygen or no oxygen, whether you're anaerobic or aerobic, glycolysis is always going to be exactly the same. Glycolysis proceeds, and at the end of glycolysis, we're going to generate 2-pyruvic acid or pyruvate. For our purposes here, they're going to mean the same thing. Remember what's produced in glycolysis with regard to ATP. There's a total of 2 ATPs produced in glycolysis, whether or not we have oxygen or not. But then, after we get to the end of glycolysis, we're in a conundrum because normally pyruvate or pyruvic acid will go into the grooming step, then the citric acid cycle, and then the electron transport chain would occur to produce a lot of energy. But we don't have oxygen, so none of those steps can happen. So instead, there's going to be another step that we can label here as fermentation. And the end result of fermentation is going to be something called lactate or lactic acid. So again, for our purposes, lactate and lactic acid are the same thing. What occurs during fermentation, the most important thing in this lactic acid fermentation, is how we can empty out the electron carriers. Remember that in order to keep glycolysis going, back up here, we have to have empty carriers, our NAD+, to come back and to glean or remove some of those hydrogens from our glucose molecule. And then we can go through glycolysis and at least make a little bit of ATP. A little bit of ATP is better than none. And in fact, while this would not sustain your life, it certainly would sustain a bacteria's life. This could be enough ATP for this bacteria to survive an anaerobic process or an anaerobic condition. And so the NAD plus is going to be here. It's going to pick up electrons, and now we have our 2 NADHs. So what occurs during fermentation is just to be able to take those NADHs and to be able to empty them back out again, so that now our empty NADH can come back and pick up more hydrogens and keep glycolysis going. If we had to say in one phrase the purpose of fermentation, it's to keep glycolysis going. And the reason why we want to keep glycolysis going is because glycolysis will at least produce for us 2 ATP molecules. That's enough to keep these bacteria alive. So the end result of lactic acid fermentation then is essentially going to be lactate. Now, let me tell you a little bit about some of the products that you might be familiar with regard to lactic acid fermentation. We know that lactic acid is an important part of how we make yogurt. When we make yogurt, we take bacteria and we place them in an anaerobic environment, and we force them to go through this process of glycolysis and lactic acid fermentation. And so when you eat yogurt, especially like a Greek yogurt, this would also be true for how we make sour cream. You know that really sharp flavor that you can get when you taste that? That's acid, and it's specifically the lactic acid. So we actually use this process of lactic acid fermentation in the process of food production for a lot of different types of food. Yogurt, sour cream, cheese making involves taking bacteria and putting them in an anaerobic environment. It's important to note that in this process of fermentation, there's no carbon dioxide liberated. So no CO2 is formed at all. So we're not going to have any CO2 released from the bacteria. That means that this lactate itself is actually going to still be a pretty energy-rich molecule. And if we find that oxygen is again restored to the system, then the bacteria can actually reverse a little bit. Then the bacteria can actually move forward with aerobic respiration and get more of the energy out of this molecule. But if there's not going to be oxygen available, then they'll continue to just take glucose, keep glycolysis going, and form this lactic acid. We'll talk about the second process of anaerobic metabolism, which is alcohol fermentation, in our very last lab of the semester.